Our topic for today is about veterinary parasitology. As in many other aspects of laboratory analysis, sample collection in parasitology is important in achieving accurate results. The sample is acceptable if proper collection is made, such as the sample should be fresh. If the samples cannot be examined within 2 hours, they should be refrigerated. As feces age, a diagnosis can be compromised, as eggs larvate and oocysts sporulate. Samples obtained later from the yard, pen, or litter box are not acceptable. The volume of sample should be adequate. A minimum of 10 grams of fresh feces should be collected. In large animal medicine, fecal samples are collected directly from an animal's rectum if possible. For the pooled samples, no one specific individual is identified. Rather, the sample represents the group. When collecting these samples, the label should clearly state the following: the owner, date and time of collection, pen number, and number of animals contained within this pen. Upon receipt of a fecal sample, a gross examination should first be performed to examine the physical characteristics. Comment on the consistency of the fecal sample, if it is soft or watery. Keep in mind that the normal consistency varies depending upon the species from which it came. Unusual color should be noted as it could give insight into other underlying conditions. For example, gray stool can indicate pancreatic insufficiency. Blood may appear bright red or black and have a tar-like consistency. Presence of mucus should also be noted. The most commonly seen parasites in dog or cat feces are roundworms and the proglottids of the tapeworms. There are many methods of preparing and performing a microscopic examination. Depending upon the suspected parasite, the sample or equipment available, one method may be preferred over another. Direct fecal smear can even be used on the small amount of fecal material. The downside to this technique is that with such a small sample, the chance of finding evidence of a parasite is greatly reduced. There are several types of fecal flotation solutions that are used as a semi-quantitative method of evaluating a fecal sample. A fecal sample is mixed with a flotation solution consisting of various salts or sugar added to water to increase its specific gravity. Parasite eggs and cysts float to the surface, while most fecal matter sinks to the bottom. Other popular solutions include Scheiter's solution and zinc sulfate. In comparison with the simple flotation method, the centrifugation technique is more efficient at recovering parasite ova from a sample. It does, however, require a little more specialized equipment. In contrast to the previous methods mentioned earlier, the Biermann technique is used to recover parasite larvae but not the ova. So this table would list the common parasites of domestic species, particularly the canine and feline. Take note that these species can be parasitized by nematodes or the roundworms. So among these are first Toxocaris species that would include the Toxocara canis, Toxocara cati, and Toxocara leonina. So these are the pictures of the Toxocara species in their egg forms. Another group of nematodes are the Ancylostoma species, which are also referred to as the hookworms. Take note that the eggs of Ancylostoma species are ovoidal in shape and are surrounded by thin, colorless shell. Another group of roundworms are the Strongyloides species, or commonly known as the threadworms. Next are the Trichuris species, or the whipworm. So this is the picture of the Trichuris species egg that is characterized by the presence of bipolar plugs. Next is the Ailurostrongylus abstrusus, or the feline lungworm. The counterpart in canine are the Philaroides species. Next are the cestodes, or the tapeworms. Among these are first the Dipylidium caninum. This is the picture showing the egg packet of Dipylidium caninum. It has several eggs which is surrounded by a thin membrane. Next are the Taenia species infecting the dogs. Another species of Taenia is Taenia taeniformis, infecting the feline. These are the pictures of Taenia species above the ova, which is characterized by radially striated shell, and the adult form below. Next are the Echinococcus species that would infect commonly the dogs. This shows the picture of Echinococcus egg, which is identical to the eggs of Taenia species. Next are the trematodes, or the flukes. First member are the Alaria species. Another important member is Paragonium kellicotti. The eggs of trematodes are characterized by the presence of operculum. Next are the protozoans. So among this is Giardia. This is the picture of Giardia cis, characterized by 4 nuclei. Next is Cystoisospora, formerly known as Isospora, which is an example of a member under subclass Coccidia. This is the picture of the oocyst of Cystoisospora felis. Another member is Toxoplasma gondii that commonly infects the cats. Mostly seen in this picture are the oocysts of Toxoplasma. The one pointed by the arrow is the oocyst of Isospora. Another coccidian is Cryptosporidium. So these are the oocysts of Cryptosporidium.